Hello everyone, and welcome back to our next lecture series in our microbiology class. This lecture is going to cover the functional anatomy of prokaryotes, so how bacteria are put together, how they work, what's inside and outside of their cells, and kind of somehow how to classify them and things like that. So let's go ahead and get started. So the first thing that we need to talk about before we dig into really what a prokaryote is what is a cell? Now, you're probably familiar with the concept of a cell already, the most simplistic form of life. They're not necessarily small, they're not necessarily big, depending on the type of cell, but they can do every single thing that life has to do to sustain itself. They can reproduce by themselves, make their own energy, and things like that. Well, every single cell, and you're probably familiar with the two different types, eukaryotic and prokaryotic. We've talked about that a little bit. Nucleus versus no nucleus, that kind of stuff. Well, every single cell, regardless of if you're eukaryotes or prokaryotes, you're going to have the same basic things in common. You're going to have the same basic shapes. All cells pretty much share the same 3 or 4 basic shapes. You're going to have the same stuff inside of your cell. It's kind of what a cell's made out of. And you're going to have DNA and chromosomes and the ability to make metabolism and things. So those are some of the traits that all cells share. But regardless of if you're eukaryotic or prokaryotic, those 3 things are the big things that all 3 cells, both of those different types of cells have. Those 3 things. There's differences between them, but those 3 things are the big ones that all cells are going to have. Same basic shapes. They're either going to be round, some sort of squarish rod shape, some sort of cube, something like that. They're all going to have a cell membrane made out of lipids, phospholipids, that's containing inside of its cytoplasm. Cytoplasm, I like to use the analogy of fruit gelatin, if you've ever seen that at a hotel, where they have the little fruit pieces floating around in the little gelatin mold. The gelatin is the stuff, the cytoplasm, and the stuff floating around the fruit is the stuff that's important inside of the cell. So the cell needs the cytoplasm, the gelatin, to keep the cell open, and for the stuff inside, the organelles and whatnot, the ribosomes in the DNA, just to not fall to the bottom of the plate if there was no jello. They just wouldn't have anywhere to float at, they just fall. And the same thing would happen if the cell didn't have any cytoplasm. The little cell membrane would just deflate, all the water would be out of the water balloon, and then all of the little fruit pieces, the organelles and things inside of the cell, would just fall to the bottom of the cell membrane and the cell would die. So cytoplasm is kind of the little liquid portion that keeps the water balloon open, if you like. And then the last one's going to be DNA or chromosomes, ribosomes, and the ability to produce energy via metabolism. So that's the same thing that both of those cell types are going to have in common. And those are the basic 3 things that all cell types, regardless of if you're eukaryotic or prokaryotic, are going to have in common with one another. Let's kind of break apart our bacteria and our eukaryotes, our prokaryotes and our eukaryotes. We've already kind of talked about this one a little bit, some of the big differences between the 2 different types of cells. Our prokaryotes, they don't have that membrane-bound nucleus. They're going to be bacteria and archaic organisms. Our eukaryotes, they have those true membrane-bound organelles, things like a nucleus, things like mitochondria, things like chloroplast. They produce all kinds of different, they're found in all different types of categories, from plants to animals to fungus, protists, so lots of different categories in the eukaryotes, whereas the prokaryotes are just going to be bacteria and archaea. So the eukaryotes have a much broader spectrum of cells and organisms found in between them, whereas the prokaryotes are more about just the bacteria and the eukaryotes. But the main differences between them are that lack of the nucleus in our prokaryotes and the presence of those membrane-bound organelles in our eukaryotes. So, once again, regardless of what type of cell you are, you're going to have to be able to carry out the characteristics of life by yourself if you're going to live. So every single cell on the planet can carry out these particular traits of life, or they have these things in common with one another. So these are a couple of more of the little traits that cells have in common. So every single cell on the planet is going to contain DNA inside of it, packed in this type of form of chromosomes or chromatin, or bacterial chromosome, nucleoid, that kind of thing. A little ball of DNA that they use to reproduce with. So either sexually or asexually. every single cell on the planet has the ability to grow and develop, or for the most part. They have to be able to get larger. They have to be able to fulfill the role that they're supposed to do. They have to allow the organism as a whole to develop, and things like that as it ages. Cells have the ability to make energy for themselves. And that's one of the big key characteristics of cells. Cells are the only things that can produce ATP, cellular gasoline. Without ATP, without cellular gasoline, the cells can't do anything. And there's nothing else on the planet that can produce cellular gasoline but cells. So cells are the only thing that can produce that cellular gasoline via the process of metabolism. Cells have the ability to respond to external or internal stimuli. They're going to read how their internal bodies are balanced and homeostasis and things like that, or if they're hungry, or if they're they need more water in, or they need to get rid of waste, things and like that. They're going to respond accordingly, or they're going to sense that it's too warm outside or too dry, and then they're going to move to a different environment and things like that to get where they need to go, to get a better environment for themselves, or to get more nutrients and things like that. So they have the ability to respond to the stimuli of the environment, and that's one thing that a rock, which is not a cell, can't do. Cells have support structures inside of them that are built for either moving them around, storing things, support, and things like that. That's kind of one of the big things of cells, regardless of which type you are. And then all cells have the ability to transport nutrients and waste in and out of their cells. So without the ability to bring in nutrients, you won't be able to sustain your life. And without the ability to get rid of the waste products that are produced by bringing in those nutrients and breaking them down via metabolism for ATP, you're going to have too much garbage build up inside of your cell, and that's going to become toxic. And then eventually you will die because you can't get rid of your waste. So the ability to bring in nutrients and get rid of waste products is unique to cells. Living organisms, rock can't do that. So that kind of idea. So these are the characteristics of life overall, regardless of what type of cell you are. So this is kind of how we break down this life of cells here again. The ability to transport your nutrients in and out of the cells, the ability to respond to internal and external stimuli, grow and develop, DNA, the ability to pass that DNA along to be able to reproduce, and then that, once again, that ability to produce cellular ATP, cellular gasoline via metabolism. So that's what makes a cell. A cell, those are the characteristics that scientists use to classify a cell. If you can't do these things, you are not a cell. You may be able to do one of them, but you can't do all of them, so that doesn't make you a cell. You have to have all of these traits to be considered a cell, or aka a living organism. So these are the characteristics of cells that all cells have in common, regardless of if you're prokaryotic or eukaryotic, and all the differences between those two. They all have these 6 in common. And these are the 6 that scientists use to classify cell as a cell. So let's go ahead and talk about a prokaryote, a bacterial cell, specifically in general, and just kind of exclude our eukaryotes for now. So we're going to start on the outside of our bacterial cell. So you can see up here the cell wall and the cell membrane right underneath it. So we're going to start on the outside of our cell and work our way in, and talk about the structures as we make our way from the outside in. So what are you going to find on the outside of a bacteria, a prokaryote? Well, the outside of a prokaryote, you can kind of break that into 2 different categories. You have the things that are used for moving or sticking onto things, appendages, things that stick out above the cell surface. And then you have the coating of the cell itself, called the Glycocalyx. And those are kind of the 2 broad categories that you can look at. So you can see here our Flagella. Those are appendages. They stick out significantly farther. They don't always have to stick out super far, but they stick out above the cell membrane and above the cell wall. And then over here, a Glycocalyx, which is attached to the cell membrane or the cell wall itself. So let's go ahead and talk about those a little bit more in detail. So let's talk about our Flagella first. You're probably familiar with a flagella if you've ever seen a sperm cell or a tadpole. They're kind of a giant sperm cell. But it's that little whip thing on the very back of a sperm cell that allows them to move around in the environment. They spin around like a boat motor. Well, flagella comes from the Latin word for whip, flagellate. So it just whips back and forth. It spins like a boat motor. And as the bacteria would be here, to have their little flagella on the back, and it spins them forward and they move in the environment like a boat motor, a little propeller. So not all bacteria have flagella. Not all bacteria have the ability to move. But if you do have a flagella, this is how it's going to be put together, depending on if you're a gram-negative organism or a gram-positive organism. Now, we'll get into the difference between those in just a second, what the actual structural differences are. But regardless of which one you are, gram-positive or gram-negative, your flagella is going to be put together the same way. So there's three main parts to a flagella. You have the filament, which is the actual long whippy part that extends outside of the bacteria that they spin around to move. It's the actual filament itself. It's a long, thin structure made out of protein called flagellin. Spins around, and that's what's actually going to do the movement of the organism. You can see that up here. It would extend significantly way up into here. And then we have the hook, which is the curved sheath that sits, and this is the bacterial cell wall. This would be the cell membrane. And this hook is going to sit inside of the cell wall or above it, depending on if you're gram-negative. So if you're gram-positive or gram-negative, it differs. So the hook kind of sits in the cell wall if you're gram-positive, and then it extends above, or if you're gram-negative, it's going to extend all the way above. Now, what the hook does is, if you can tell here, it's kind of curved. It has a little slant to it, and it makes the flagella spin in a circle. If the hook was straight up and down and it just spun like this, the flagella would just do this, and the organism would not be able to move. So it's kind of hooked like this. It's got a slant to it. So as it spins, it causes the flagella to spin in a circle. It's the same concept of propellers are kind of slanted a little bit. If they were perfectly flat, you'd have no propulsion, so they have to be slanted a little bit. And that's the same concept here. It allows it to spin around instead of just spin in a flat line and not actually cause any propulsion. So that's what that's for. And then you have the anchors, which are actually going to hold the flagella and the hook to the cell. And those are called basal bodies. So if you've got a rope and you just throw your rope out into the ocean, it doesn't really do a whole lot. You have to tie it to the boat to hold the boat there. And that's what the anchor is going to do. So the basal body is going to hold the giant rope that they use to swim around with to their cells, hold it in place so when it starts to spin around, it doesn't just float off into the abyss. Now, if you're a gram-positive organism, you have one set of basal bodies. And if you're a gram-negative organism, you have two sets of rings and one set of basal bodies. So one set of basal bodies, two sets of rings, one set basal body over here with one set of rings. So and we'll talk about why this is different in just a second. So what does a flagella do? I've mentioned it rotates in a circle, and it allows the bacteria to move around in their environment. Now, these guys work a lot like boats. If you've ever driven a boat, or a car for that matter, and you just let off the gas pedal or let off the throttle on the boat, they don't come to an instantaneous stop. Boats are even worse about it because of the less friction in the water. And these guys kind of work more similar to boats than cars. Or more similar to airplanes, if you like, but let's stick it to the boats for now. So anyway, you turn on the throttle in a boat, you go really fast, and all of a sudden you just turn off the throttle, and the boat doesn't just instantaneously stop, and neither do bacteria. They're going to turn their flagella on, it's going to spin really fast, they're going to run forward towards food, towards light if they're photosynthetic, or away from something toxic, or away from the light if it's too much light. They're going to move around to where they want to go. And when they get really close to where they're going to want to go, they're going to turn off the boat throttle. They're going to turn their flagella off, make it quit spinning. Well, most of these guys live in some sort of semi-aquatic environment that allows them to move around, so they float. They swim around through the environment. So much like a boat, they get close to where they want, they turn their gas pedal off, and then they just kind of tumble around into the environment until they run out of momentum, and they just kind of naturally come to a stop, very close to where they want to be. Or at least that's the hope. So the way that bacteria move is called run and tumble. They spin their flagella, they run forward, get really close to where they want to be, and then they turn their flagella off, stop the gas, and then they just kind of tumble to a stop as they run out of momentum where they want to be, or at least hopefully where they want to be. And that's what the function of a flagella is. That's how it works. Now you can see down here an organism with one flagella. You can see it just spins its single flagella, moves around very easily. Now, if you're an organism that has a bunch of flagella, they're going to all collectively spin in the same direction, which will collectively cause them to come together into one kind of giant super flagella. Kind of like braiding. If you've ever twisted stuff together, it just twists together and gets tighter. And that's what happens to these flagella as the organism spins around. spins the flagella, they all twist together into one big super flagella that can power the organism. As they run, they get to where they want to go, they stop, and then all the flagella just kind of untwist and go back into their natural position around the organism. So this is the run and tumbling movement of organisms. Now, this is a really good way to break apart different species of bacteria, or to identify different species of bacteria underneath a microscope, is the way that their flagella are organized around their bodies. Now, as I mentioned, not every organism has a flagella, but if you do have a flagella, these are some of the really common arrangements for flagella that are found in organisms. You have something called monotrichous. Now, mono meaning one, trichous is the word for hair or thread. So one singular hair. So this is a monotrichous organism. It has one flagella coming out of one end of the organism. That organism's the very classical sperm cell kind of movement, just runs around in one little direction, gets to where it wants to go. Now, I've been comparing the sperm cell to these with this flagella, and the only thing that eukaryotes and prokaryotes share in common in the flagella is the way that it moves. They're not put together similarly at all. They're not related to one another. It's just the way that they move. It's kind of like a jet engine and a propeller. They both allow the airplane to fly, but the way that they do so is functionally totally different. So the eukaryotic and the prokaryotic flagella sperm cells, eukaryotes, these guys are prokaryotes that we're talking about here, they're not related to one another. They're not they're not similar at all. They just do the same thing. They allow the organism to move, and that's pretty much it. So I should clarify that before we get too far into this. So anyway, so monotrichous, mono meaning one, trichous meaning thread. So one thread coming out of the end of the organism, and that's what they're going to use to swim around in the environment. You have lopotrichous, which is a small bunch of flagella coming out of one end of the organism. So you can see here, this is a spirillum, a very interesting little shaped organism, look kind of like a pasta noodle. And they have flagella that come out of one bunch down here, a bunch of them, that come out of one spot. You can see 6 or 7, maybe 8 or 9 of them, all coming out of the one end of that organism. Now, as that organism is going to move, it's going to twist all these flagella together into one superflagella, and that allows them to move forward. And that's a lopotrichous arrangement, a bunch of flagella coming out of one spot. Now, amphitrichous, this organism has flagella at both ends of the cell. So it looks like a tic-tac, and you have flagella at either end of the tic-tac, little bunches. So this guy would be on either end of the cell. So one over here and another little bunch over there, amphitrichous. And then peritrichous, so peri means above or around. So hair all the way above or around this organism. And that's one of the characteristics of what we talked about here, with that weird, where all the flagella are going to twist together around the organism into that one superflagella to allow it to move forward. So monotrichous, lopotrichous, amphitrichous, and peritrichous. I'm going to come back to that in just a second. But there's one more type of flagella arrangement called a periplasmic arrangement. Now, we'll get into the shapes of bacteria and things in a little bit, but there's two main different shapes of bacteria that are spirally. You have a spirillum and a spirochete. Spirillums and spirochetes, they look very similar underneath the microscope, except for the fact that spirillums have external flagellae. So this is a spirillum right here. You can see its flagella. They're external. Spirochetes, on the other hand, have internal flagella. Let's get back here to the breakdown of our cell membrane, cell walls. It's going to sit in between that space here. So it's not going to be above the flagella. It's going to be stuck in between their cell walls. So you can see that here. So this is the cell wall of the organism, and the flagella sit right around that. Very interesting how this works. They're kind of embedded in that cell wall. They're hidden from the environment, so they don't stick up in the environment. They're not poking out of the cell. Now, how this works is it allows the organism to twist up kind of like a rubber band. So the organism is going to twist those flagella inside of it, and it's going to cause a lot of pressure inside, and then they're going to let go of it, and it's going to spin them forward like a rubber band wind-up car. And this is essentially how these organisms move around in the environment. Very strange. So that's how you tell apart a spirillum from a spirochete, those internal versus the external flagella. And that's what you're looking for here. Spirochetes have external, spirillum have internal. I got that backwards. Spirochetes have internal flagella, spirillum have external flagella. And that's the difference between the two. So another thing that's found on the outside of prokaryotic organisms are called fimbriae. Now, fimbriae are very interesting little bristles. They're made out of protein. They're very thin, very small, that extend just a little bit above the cell wall, cell membranes of these organisms. And what they do is they're essentially just there to allow the organism to kind of work like Velcro. They just stick to the environment. So these little fimbriae are sticky, and you can see down here that they're going to allow, these are E. coli that's found in our intestines. E. coli play a huge role in our intestines. We'll talk about this later in this course, regulating the body. They play a huge role in the immune system, things like that. And they line the insides of our intestines. Now, as the food passes through our intestines, water and things like that, these E. coli are going to be pushed through the intestines. Well, if they're all pushed through, they're going to be pushed out. They're not going to be able to survive there. They naturally live there. We need them. That's a good place for them to be. So we don't want to get rid of them. So how do they stay in place? Well, they're going to use these little fimbriae that are all around their cells to stick to the cells of our intestines. So these fimbriae are going to be used to stick bacteria, prokaryotic organisms, in place, to hold them to surfaces, to make sure that food doesn't push them out, that they don't get washed away and things like that, to make sure that they stick where they want to stay. And that's what fimbriae are used for. Another thing that's found on the outside of cells are things called pili. Now this is a pili here, you can see it as well. Now what is a pili? Well, this is the Greek word for stick or spear. And essentially this is a reproductive structure that's found only in gram-negative cells. So gram-positives don't produce these, only gram-negatives. And what it does is it kind of allows these organisms to have a one-way exchange of genetic information. So one organism will produce this pili. You can see this one here has produced it. This organism is incapable of producing a pili. We'll talk about this process later. It's called conjugation. And this pili is going to extend from the organism that produces it to the organism that's going to be receiving the genetic information. It's essentially like a big, giant, long, hollow protein straw. So this organism will produce it. It'll stab it through the cell wall, cell membrane of this other cell, and then some plasmids or some DNA might be passed between this cell to that cell. And then this cell will be able to use whatever information that this cell gave it through the sexual pili. And that's what this little thing functions as. It's a straw that allows one organism to give genetic information to another. And that's what they're used for. Pretty interesting process. Now the other thing that's found on the outside of bacteria and prokaryotic organisms is a glycocalyx. Now you will find these in some eukaryotes, things like yeast. But in bacteria, they're going to be a little more common, and that's going to be used a lot more for diagnostic purposes if you're looking at it on a petri dish. There's two main types of a glycocalyx. Now, a glycocalyx is just a bunch of sugar and protein that's going to surround the outside of a cell. Now, if you recall, I mentioned heat fixing earlier in this lecture series. That coating that kind of gets sticky and melty and stuff, and this is what that is. That's going to allow them to stick to the slides. That sugary, gross layer that's going to stick to the cells, stick the cells to the glass slides. This is what that is. Anyway, there's two different forms of that. You have something called a slime layer and a capsule. Now, if you recall a capsule from earlier, we talked about capsule staining, is that really thick structure that's going to surround a bacteria that keeps it from being phagocytized by the immune system. So a white blood cell comes up and tries to eat it, and it's going to pass right through that cell, or the cell won't be able to eat it, and then that bacteria will be completely unharmed and completely able to continue disease and things like that. So that's what a capsule is. Well, a capsule is a really highly organized layer of very tightly compacted proteins, very tightly compacted sugars that are very closely held together, very tightly compacted around the cell itself. They're very highly organized, they're very highly structured, and it's kind of like a snowball. You go outside and you pick up a snowball and you crush it all together to make a really nice snowball. And that's what a capsule is, very hard, tightly packed blob of proteins and sugars that offers a lot of protection to the cell. And the other form of glycocalyx is a slime layer. And this is you go outside and you pick up a bunch of snow and you just kind of do this a little bit with it, and it's kind of going to fall off a little bit. It might stay in place a little bit, but it oozes, it kind of slobs around, and it's going to fall off, and it's not going to stay where you put it in the nice shape of a ball. So a slime layer is not very tightly organized. It's not very tightly packed. It kind of sloshes around. It oozes around the surface of that organism. So you can tell them apart on a petri dish if you look at them. This is a slime layer. It's significantly more oozy looking. It's kind of not very organized and things like that. Very blobby looking. Whereas you can see over here these really nicely, tightly compact, very shiny looking, very round colonies that have capsules around them because everything's highly organized together. So this is a slime layer versus capsules on a petri dish, and that's what this is the difference between the two on a chemical level. So regardless of what type of glycocalyx you have, a capsid, a slime layer, or a biofilm, it's going to do these three things. It's going to keep you from being eaten by the immune system. So you can see here our little macrophage or our white blood cell is going to come up. It's going to try to eat our little organism with the capsule around it. And it just can't fit it in its mouth. It's unable to phagocytize that organism, which means that it won't be able to kill that organism, so that organism will be able to continue to cause disease in the host, potentially leading to death. So that's one of the big things that it does. If you have a capsule or a slime layer, you are significantly better at causing disease than organisms that don't, even if you're the same species. It's also going to keep you from getting dried out in the environment. It's also going to help retain moisture and nutrients, and that's kind of the big deal here. It would be kind of like wearing a raincoat over you. If you sweat, all of the water is going to get trapped inside of the raincoat, and then it's just going to condense, and you can potentially get the water back. And that's the same way for these guys. It protects them from drying out. Any moisture that's going to be lost is going to be protected inside of the capsid. It keeps nutrients from being lost to the environment and things like that. And it also allows them to stick to stuff. So capsids and slime layers are sticky. And the biofilms that we talked about previously, how they start with the snowball concept, one little bacteria sticks to the surface and then another one sticks to that, and then they build up over one another. They're going to stick with that glycocalyx, and that's what's going to stick, that little sticky, sugary coating. It's going to stick to something, and then another bacteria is going to stick to that. They're with their slime layer or capsid, their glycocalyx, and another one's going to stick, and then so on and so forth, and they're going to build up on top of one another to form a giant biofilm, all connected by their giant connected slime layer at that point, or giant connected capsid and things like that. So very odd things that these organisms are capable of doing once they connect to one another. So we've talked about biofilms already. That quorum sensing, these ability for these cells to talk to one another. You can see them here. This is the surface of a toothbrush. And you can see here, I think this is a toothbrush bristle that they zoomed in on. And you can see the actual bacteria that are stuck to that bristle in a biofilm. They're all big blobbed over the surface. You can see them over here and things like that are all stuck together on the surface of that toothbrush bristle. So now let's take our way from the outside of the cell. We've talked about the things that are on the outside, the flagella, the fimbriae, things like that. And now let's move on to the inside of the cell. So we're not going to break through onto the cytoplasm and things like that. We're going to talk about the cell envelope. And this is right underneath our flagella, what they're going to be stuck in and things like that. So the cell envelope is going to contain two big things, our cell wall and our cell membrane. Now, every single cell on the planet, regardless of if you're prokaryotic, eukaryotic, gram-positive, gram-negative, whatever, you're going to have a cell membrane. That makes a cell a cell. Now, bacteria, they have a cell membrane, but almost all of them are going to have a cell wall as well. So the cell wall and the cell membrane, collectively, are referred to as the cell envelope, the external coating of the cell that contains the cytoplasm on the inside. And that's what the cell envelope is, the external coating. So inside of a bacteria, you've got that cytoplasm, and that's what's going to keep the stuff floating around. And on the outside, you got the cell envelope, the cell wall, and the cell membrane. And the cell wall and the cell membrane are going to protect everything on the inside. They're going to keep stuff safe on the inside and make sure that the cell's not punctured or broken. And it also keeps water pressure regulated on the inside of the cell and things like that. Just make sure that the cell stays as it should. Now, if you break apart bacteria, just bacteria, we can look at their cell walls, the difference between the cell wall, not the cell envelope as a whole, the cell walls, using something called a Gram stain. Now, we've talked about a Gram stain already. It's used to break apart cells bacterial cells based upon how their cell membranes are put together, their cell wall, sorry, not cell membrane, the cell walls. And this is how we're going to break apart these types of bacteria into a gram-negative classification or a gram-positive classification. And I've mentioned these terms before, but now we're going to talk about what they actually mean. Now, organisms that are gram-positive bacteria, they're going to have a very, very, very thick layer of peptidoglycan that's found inside of their cell wall. They're going to have one cell membrane, and then they're going to have one cell wall. So you can see here, this is a gram-positive organism. This is their one cell membrane, and then right above that, they're going to have one really thick layer of peptidoglycan. Now, I've talked about peptidoglycan before. It's the the sugar protein structures that are found only in prokaryotes. It's the thing that makes up their cell membrane, cell walls. Only found in prokaryotes, not in eukaryotes. So these guys, these gram-positive organisms, have a very, very, very thick layer of peptidoglycan and one cell membrane. Gram-negatives, on the other hand, they're going to have one cell membrane, one very, very, very thin layer of peptidoglycan cell wall, and then another cell membrane that sits right above that cell wall. So gram-positives have one cell membrane, one very thick layer of peptidoglycan. Gram-negative cells have one cell membrane, one very thin layer of peptidoglycan, and then another cell membrane that sits above that peptidoglycan cell wall called the outer membrane. So they have the outer and the inner, and then a very thin layer of peptidoglycan. So that's the difference between a gram-negative and a gram-positive cell wall. So now let's dig into that a little bit more. So, peptidoglycan. What is peptidoglycan? Well, peptidoglycan is a structure that's found only in prokaryotes. That's put together of little tiny sugars and little tiny proteins that are linked together in a very unique structure. It's kind of like a chain link fence. It's kind of how they're put together. And this way that the peptidoglycan is formed is very unique, very rigid, very controlled, and a lot of drugs prey upon this. So imagine having your chain link fence and then something like penicillin to prevent you from being able to form half the fence. So cell walls, as I mentioned, are meant to keep the cell from being able to pop from water pressure and being able to keep things safe on the inside. So if half of your chain link fence is missing, you're not going to be able to retain a lot of water pressure. You're not going to be able to sustain a lot of damage if you're missing half of your fence. So that's how a lot of drugs work. They break apart peptidoglycan, so the bacteria lose the ability to maintain water pressure and things like that on the inside, so they just pop. Now, peptidoglycan, as I mentioned, is only found in our prokaryotes, and our gram-positives have a lot of it. They have one cell membrane, their cytoplasm and everything's on the inside, same as a gram-negative on the inside, pretty much. But they have that structural difference on the cell wall, and that's what makes them different. That cell membrane and that one thick layer of peptidoglycan. Now, that one thick layer of peptidoglycan is going to make these guys very resistant to water loss. These gram-positives are going to be found a lot in the environment or on your skin. They're good at maintaining water pressure on the inside of them because that very thick cell membrane, very thick cell wall can maintain a lot of water pressure, a lot of damage. So these guys are very hardy organisms. They live a lot in the environment and on our skin, exposed to air and things like that, because they're very resistant to drying out, so desiccation. That's the thing that bacteria have to worry about a lot, just losing too much water to the environment. Another thing that's going to be found in gram-positives, that's only found in gram-positives, is teichoic and lipoteichoic acids. Now, these things are going to be bound inside of that cell membrane, or sorry, that cell wall, that peptidoglycan cell wall. Now, I've got a three-dimensional picture of this in a little bit, so don't worry about it, you'll be able to see this. But these two acids, these two things, function to kind of regulate the cell membrane and cell walls during division of the cell to make sure that the cell wall is being made properly. It also helps move things across that cell wall. You have a very thick cell wall. It's hard to get things across a very thick wall. So they're going to help regulate the movement of some things across the cell envelope. And sometimes they can trigger an immune response in patients depending on what type of bacteria and things that they are. Now you can kind of see here, not every single bacteria, not every single prokaryote has this, but these do. In between the cell membrane and the cell wall here is this little space, and this is called the periplasmic space. Peri meaning above or around. Again, same thing as that periplasmic flagella. So above or around the plasma membrane. So it sits right above the plasma membrane, that little tiny space. Now, bacteria, if you have a periplasmic flagella, your flagella will sit there, and if you're a spirillum. Or if you're not, and you're just a regular old bacteria, that's where your hydrogen ions are going to be pumped for your electron transport chain. So this is essentially a mitochondria, and this is the space inside of a mitochondria that holds the hydrogen ions. So this is essentially what that functions as. And they can also store some nutrients and other things there. So not every bacteria has it. Not every single gram-positive has one, but this one just happens to. And on to our gram-negatives. So you can see here our gram-negative, that one cell membrane, cytoplasm and stuff on the inside, one cell membrane on the inside, very, very, very thin layer of peptidoglycan, and one outer membrane on the outside above that. Now, this organism has two periplasmic spaces, one that sits above and one that sits below the cell wall, that they can use for storing nutrients, storing their hydrogen ions, and things like that. So, this structural difference right here, I've mentioned earlier that gram-positive drugs and gram-negative drugs tend to not work on the other classification of bacteria, and this tends to be why. If you have a drug like penicillin that targets the peptidoglycan cell wall, peptidoglycan on a gram-positive is exposed to the environment. Peptidoglycan comes in contact with penicillin. It's very easy for the penicillin to go to work. Now over here, penicillin can't get to the peptidoglycan. It's hidden by the outer membrane. So the outer membrane protects the peptidoglycan from the penicillin underneath it. So no, this guy could bathe no matter how much penicillin you pour on it. It's not going to do anything because it can't get to the peptidoglycan layer to cause harm to this organism. So that's one of the reasons why drugs that tend to work on one class don't work on the other. And that's due to the structural anatomy of the cells. There's a bunch of different other reasons, but that's a really big one right there. It has a hard time getting to the targets on the other classifications of the drugs. So once again, this is the classification for gram-negative, how their cell envelope is put together. Not all of them have two periplasmic spaces, so keep that in mind as well. Another thing that's going to be found, a big one here in our gram-negatives, only in our gram-negatives, is something called lipopolysaccharide, or LPS. Lipo meaning fats, polysaccharides meaning starches and sugars and things like that. So it's a protein, it's a fat and a sugar put together, lipopolysaccharide. These are also known as endotoxins. Now, endotoxins, lipopolysaccharide, cause something called toxic shock syndrome. Lots of streptococcus organisms and things can cause this. They can cause this, but not the same way. E. coli and things like that can cause it through this particular means. But toxic shock syndromes tend to be caused a lot by LPSs from organisms that are gram-negatives, like E. coli and things. This is a big deal for lots of clinical babies tend to get this a lot. toxic shock syndrome and things like that. So once again, I've got a picture of LPSs, and you can see how these are bound inside of the cell wall. But endotoxins and exotoxins are a topic for later, and I'll kind of briefly touch on that in just a second. So here's that 3D picture that I mentioned. You can see them put side by side here. Our gram-positive over here, one cell cell membrane, very thick cell wall of peptidoglycan with teichoic and lipoteichoic acid bound in it. Over here we have our gram-negatives, one cell membrane, one very thin layer of peptidoglycan, and our outer cell membrane with those LPSs bound in it. Now, when cells die, they're going to rot, they're going to break apart, same as every other living organism on the planet. And when these cell membranes break apart, these lipids, these LPSs, are released from the cell membrane, and then they can start to cause problems, and that's what toxic shock syndrome is. You have a bunch of bacteria in your body that die all at once. They all instantaneously release their LPSs, and your body's overwhelmed by all of those LPSs in the body, and the immune system can't handle it, and it goes into just kind of shock, overdrive. And that's what toxic shock syndrome is. Now, an endotoxin versus an exotoxin. Endotoxins are inside the cell, and they're only going to cause problems once that cell dies. Lipopolysaccharides, since they're embedded inside the cell membrane, they can't cause you problems if that cell is living. Once this organism dies, however, and starts to rot and fall apart, these LPSs are going to be released into your body, and that's when they can start to cause problems. Exotoxins, on the other hand, are going to be released externally of the cell, and a living organism doesn't even have to be present. You just have to come in contact with the toxin for it to cause problems. So endotoxin versus exotoxin, we'll get into that later in greater detail, but that's kind of what the idea is for now. So there's a couple of more differences between them. You can check those out, dig into them, the differences between a gram-positive versus a gram-negative cell walls. So there are a couple bacteria that don't necessarily have cell walls, or cell walls in the stereotypical sense. Now, we've talked about cell walls that have mycolic acid bound in them, those organisms that are acid-fast staining. That mycolic acid that's bound inside of their cell walls allows them to be very resistant to drying out in the environment's waxy structure. They don't stain. Any type of stain's going to wash off them. They're very resistant to water loss and chemicals and things like that. So that waxy coating is going to be found inside of their cell wall. Now, if you try to give these a gram-positive or gram-negative a gram stain, they would stain gram-positive, except for the fact that that mycolic acid prevents them from staining at all. So use mycolic acid, you have to bind something that binds to that. So that's why that acid-fast concept comes into play. They are technically gram-positive. They have that big thick layer of peptidoglycan, one cell membrane, but that mycolic acid that's found inside of their cell envelope prevents them from staining gram-positive because you can't stick crystal violet to it. And then there are some organisms that have no cell walls. If they have a structure that kind of resembles a cell wall, but it's stabilized by sterols. It's not peptidoglycan. So a very interesting way of doing this. They don't have shape. So cell walls define the shape of the organism. It's kind of like once they're made, they're stuck in that shape. Once the chain link fence is put up, it's not going to change shape anymore, and that's essentially how they work. Once the cage is built, the organism lives inside the cage. Now, if they don't have a cage, no cell wall, they have no shape. So these organisms can change their shape. They're called pleomorphic, many shapes. So you can see down here, these are pleomorphic organisms. Mycoplasma causes walking pneumonia. And you can see down here, this is all the same organism. They just don't have a set shape. Some of them are round, some of them are kind of long and stringy, some of them are circular, caucus shaped, some of them are just kind of weird. They don't have a set shape because they have no cell walls that are hard and rigid. They can flex and shape and move around and ooze kind of like an amoeba. So organisms that don't have one set shape make it very difficult to diagnose, very difficult to stain. You mostly have to diagnose these types of infections based on DNA presence or antibody tests and things like that, because you really can't see them underneath microscopes. They don't stain, they're very hard to test for, and things like that. So very interesting little organisms, and they lack their cell walls. So let's talk about how the cell membrane's put together. You're probably familiar with this one already. This is called the fluid mosaic model. You've seen the plasma membranes. We've talked about the phospholipids that make them up, the saturated versus the unsaturated, the ones that kink in them. The saturated and unsaturated are put together in certain ways that allow the cell membrane to be kind of fluid. It floats around. The stuff on the surface moves around, floats around. The plasma membrane, the phospholipids can shift around, and kind of like a surface of a lake. While the water doesn't go anywhere, it's always moving, and that's kind of the same thing that's going on the surface of a cell. Everything's moving around, even though the cell's still there. Now, what the cell membrane does is it's going to do lots of things. It's going to keep everything inside the cell. It's kind of the membrane, the water balloon, if you like, that keeps the water inside the balloon, the latex part. It's going to allow certain things in and out of the cell selectively. That's one of the big key parts of cells. Their cell membranes are selectively permeable. Certain things can come in that the cell likes. Certain things can go out that the cell doesn't want to, and the cell can determine for the most part what is going in and what's going out, so it has control over that, so it's not just killed by stuff just randomly floating in. And then the cell membrane is going to allow a space for the cell to do things. You need attachment points inside of a cell, kind of like a wall. You can't just hang a picture floating up on in the air. You have to have a wall to hang the picture to, and the cell membrane is kind of the wall for a cell. All the proteins that they need to do things, they'll hang up on the cell membrane. Stuff that goes in and out of the cell, they'll hang up there to get stuff through in and out. They'll hang up little information that says, hey, I'm a healthy cell, hey, I'm ready to reproduce on the outsides of their cells so other cells can read them. All kinds of stuff that the cell membrane does. So very, very, very cool, very interesting functional thing on the outside of cells. So let's go ahead and move on to the inside of a cell. So we're talking about our prokaryotes again. But regardless of if you're prokaryotes or eukaryotes, you're going to have cytoplasm. That's our key characteristic of all cells, that jelly-like substance inside of the cell. It allows the cell to have space. It just allows them to have stuff inside that stuff can float around and move around inside of. You need water for the icebergs to float around in, and no water, the icebergs just sink to the bottom of the ocean, and that's what would happen inside of the cell. It would just float down, all the cell membrane would sink, all the little organelles and stuff would fall at the bottom of the cell, and it would just die. So very unfortunate without cytoplasm. Cytoplasm is basically just water with a bunch of sugars, some salts, and some other stuff dissolved inside of it that just allows for the cell to move stuff around in. That's basically what it does. Some other stuff you might find inside of the cell of a prokaryote is a nucleoid, a singular, circular, double-stranded blob of DNA. It's just a ball that's stuck together. It's kind of like a yarn ball of DNA. There is no membrane around it. There is no nucleolus in the middle. It's just a yarn ball of DNA, naked little yarn ball. And that's going to be that everything that bacteria needs to make a copy of itself. When that bacteria comes time to reproduce via binary fission, it will copy this little nucleoid or this bacterial chromosome and make a new copy of it, and then one side will go over to one side of the cell and the other, and that cell will split in half and make two identical copies. So that's what a nucleoid is. It's everything that cell needs to make a new copy of itself. Now what you also might find inside of a bacterial cell or prokaryote is something called a plasmid. Now not every bacteria is going to have them. They don't need these things. It's kind of like a travel guide. You don't need a travel guide to go to a foreign country, but if you have one, it's kind of nice. They know some things that you might not know, and they can teach you some tips and tricks that you might not know either. So plasmids work kind of like this. They are little tiny circular balls of DNA that are going to be passed from bacteria to bacteria via those peleases. They can also get them from the environment and things like that. They can bring them in. But they don't have to have them to survive. It's that concept. You don't have to have a travel guide, but they're nice too. So bacteria do not need these plasmids to survive. But if they get one, either from the environment via conjugation, or maybe we give them one from the environment, or from a genetic manipulation lab or something, they can usually use those plasmids for some really cool stuff. So the information that's contained on that little circular ball of DNA is going to allow that organism to now make a brand new protein that can allow it to eat a new nutrient source. Or maybe it's going to produce penicillinase, an enzyme that breaks down penicillin. So now that once susceptible gram-positive is now immune to penicillin. You can also use plasmids in a laboratory to genetically manipulate cells. We can put information that we like inside of a plasmid and then stick it inside of a bacteria and make them do things for us that they wouldn't normally do. So plasmids are super cool. Once again, not required for a bacteria to live or to function, but you might find some inside of some organisms. What you will also find inside of the bacteria, prokaryote, is a 70S ribosome. Works the exact same as a eukaryotic ribosome, it's just slightly smaller. It's where proteins are going to be made, and every single cell on the planet, prokaryote or eukaryote, has ribosomes. It just depends on the size. And once again, our prokaryotes have a 70S that's slightly smaller ribosome than our 80S found in our eukaryotes. You might also find some just junk inside of the bacteria called inclusions or granules. And bacteria may or may not have these. They vary in size and shape and number. Even what's inside of them is going to vary. And this is when a bacteria comes in contact with something that it likes in the environment, or that it needs a lot of. So an organism might require a lot of iron, a specific organism, in its metabolism, things like strep. So if this particular strep organism comes in contact with a bunch of iron, it might overeat or take in way more iron than it needs to and store it inside of its cell. You can see them here. There's no telling what's inside of these. These may be nutrients, these may be metals, it could be anything. And then once that organism has used up all of the whatever that resource is in the environment, it can use the granules inside of its body for the source of those nutrients. So when the environment's exhausted, it still has some saved over. And you might find these inside of some organisms as well. On the inside of a bacteria, you're also going to find the cytoskeleton. And this functions the same way as our skeleton. It essentially just provides support for the inside of the bacteria. Now, it sits right underneath the cell membrane. It's kind of in between the cell membrane and the cell wall. And it's just kind of a little curly queue of connected actin filaments that expand and sit. It's kind of like a spring. If you've ever seen one of those little dog toys that are like a little slinky toy, like the magicians use around, it's got the slinky on the inside of it, like a little sock tube, and it slinks around like that and it moves around like a little animal. And this is essentially what that is, a little spring on the inside that adds a little bit of rigidity to the cell itself. The cell can also use these little actin filaments to move things around for lots of different purposes, but it's mainly just meant there for support to the cell structure itself. Endospores, we've talked about endospores a lot. Some of the other things that you might find on the inside of a prokaryote. These inert resting stages of a bacteria that are very highly resistant to death. The only way to pretty much kill them is to burn them. They're not going to die from boiling water, from freezing, or things like that. They're very tough and allows bacteria to survive for an extremely long time in very harsh conditions. And then they come back to life. That metabolically inactive endospore can pretty much sit there forever. And then once the environment becomes better, the cell can become active again and start reproducing and start a new colony for itself. Now, these endospores, once again, are only found in some gram-positive rods, but they're only produced by gram-positive rods, but only some of them produce them. So Clostridium, Bacillus, Sporosina, or another one. Those are some of the very common genuses that produce them from the gram-positives. Now, there are if you can produce endospores, the endospores will be produced in something called the sporulation cycle. And that's the formation of an endospore. If you're not forming an endospore, you are considered a vegetative cell. You are a metabolically active, growing, happy cell doing your thing, chilling out, existing. And then once the environment starts to go bad, this organism will enter into the sporulation cycle. Gram-positive rods, once again, are the only cells that can do this. Gram-negatives, when the environment goes bad, and gram-positives that can't do this just die. So organisms that can do this will do this. If you can't, you just die. So the vegetative cell will essentially quarantine off a little small section of its DNA. It replicates its nucleoid, quarantines off a little section of its nucleoid or that replicated piece of its nucleoid in the front of its cell, starts to pack it up with all of those protective proteins, protective fats, protective sugars, starts to disintegrate the other part of the cell. The original part of the cell will break down the DNA, break down the cell membrane as the environment starts to deplete. And then essentially you're left with an everlasting gobstopper surrounding a jawbreaker, surrounding a sleeping little bacteria that's in hibernation forever. And then once the environment gets better, this endospore now, so this is the vegetative cell, sporulation cycle, formed our endospore. The endospore will start to rehydrate, start to get some nutrients and wake up essentially, and now it will start to become a vegetative cell again. So you're either going to be a vegetative cell in the sporulation cycle or an endospore, and that's the three cell states that are found in cells that can produce endospores. So we've talked about endospores. They've been around for a really long time. They found endospores in Arctic ice cores that date back to 250 million years, if not a little older, and they've been able to revive them successfully in a lab. So very strange. About the only way to destroy them is to burn them or to use pressurized steam, and that's a really good way. So the autoclave will kill them. So let's go ahead and talk about bacterial shapes. I've mentioned the gram-positives, the gram-negatives, and things like that. So regardless of if you're gram-positive or gram-negative, you're going to be pretty much one of these three shapes. These are the three main shapes of prokaryotes and bacteria overall, and they're going to be one of these three main ones. You're going to be a coccus, which is the Greek word for grape, a bacillus, or a rod, depending on the type of species or book that you're using. I prefer using the term rod. Bacillus is also a species of bacteria, so if you recall earlier I mentioned why it's so important to clarify if you're writing, to italicize, to write the name properly, because if I say bacillus, I could be talking about the shape or the species. So you have to clarify when you're writing, because that gets a little overly innocuous, especially when you can't ask someone questions. Or you're going to be a spirillum, a spiral. Now remember, we have our spirillums, which are going to either be spirochetes or spirillum, which our spirillum have those external flagella and our spirochetes do not. So that's the difference between those. So you're either going to be a nice little coccus, a rod, or a pasta noodle, a spiral-shaped bacteria. Now, there are some bacteria that kind of don't lack shape, that lack that shape, like I mentioned earlier. They have those weird cell walls that don't necessarily have a shape. These guys, these coccuses, rods, and spirillums, they have a set shape by their cell walls. Their cell walls form. They're set that way that bacteria is not going to change shape unless it's damaged. These guys have no set shape, so they are going to look different underneath a microscope when you look at them. So you can see here, these guys, this is diphtheria, the bacteria that causes diphtheria. And you can see here that they are slightly different shaped. They're not the consistent little rods, consistent little circles like these guys are. They're all the same shape, kind of things like that. So pleomorphic bacteria, let me go back the other way. Pleomorphic bacteria are interesting. Now, another thing I've mentioned a lot when it comes to staining is you get the shape of an organism. And those are the shapes. Our coccuses, our rods, or our spirals, one of those three shapes. And you get the arrangement. And this is another thing that you get from a stain. So the arrangement of a bacteria refers to the shape and how those shapes are put together. So if you've got more than one cell, how are those cells going to be arranged? Well, if you're a coccus, you're either going to be a single coccus by yourself, a diplococcus, tetrads, 1, 2, 3, chains, 1, 2, 3, 4, 5, 6, little long chains, or clusters. And that bit of information can tell you a lot about an organism. So you can be a singular coccus, like here. You can be two cells put together. You can see those here, one cell, and then one cell, one cell. So diplococcus, two little circles put together. A staphylo arrangement, and this means cluster. Staphylo means cluster. Coccus means grape, so cluster of grapes. Staphylococci, a ball of circles. And this is once again why that concept of Staphylococcus as the species, italicizing it, or just writing it normally comes into play here. This does not mean the species Staphylococcus that causes staph infections. This just refers to how these organisms are arranged. If it was italicized, it would be the particular species, Staphylococcus, that's also arranged this way, which is where the name comes from. But there are lots of organisms that are not staph that are arranged this way. So this is just the arrangement. You have Streptococcus. Strep is the Greek word for chain, so chain of circles. So chain of circles, long line of chains, line of circles. So Streptococcus bacteria are obviously put together this way. Once again, where that name comes into big play, there are lots of chains, circles that are not Streptococcus species, so big deal there. Make sure you're writing it properly. Sarcina, little 8 of them put together in 4 circles, and then a tetrad, so 4. So those are some of the common arrangements that you'll find in bacteria. Now, if you're a rod, it's pretty much the same. You're going to be a bacillus or a rod, a diplorod, a diplobacillus, streptobacillus, streptorods, kind of things like that. So pretty much the same terms are going to apply on the arrangements. It just depends on if you're a coccus or a rod. And then you have some variations on the central themes. You have a vibrio, which is kind of a curved rod. You have just the spirochetes and the spirillums, the spirally bacteria. And then you just kind of have the pleomorphs, which are just the others, the random forms that are out there. So these are some of the very common forms of bacteria that you'll more than likely encounter all the time in a medical lab. So once again, the arrangement here, something to keep in mind as well. So bacteria shape you get from the stain. Bacteria arrangement you get from a stain, and that's what you get from a simple stain as well as the size. And then you get the gram stain reaction from our gram stain. And that's what you get from a gram stain that you don't get from a simple stain. And now you know what that gram stain reaction has to do with how the cell walls are put together. And that also allows you to start treatment quicker because you know if you have a gram negative, you need to get this type of antibiotic, or gram positive, and so on and so forth. So now that you know all this stuff about how a bacteria is put together, what it looks like, what the shape of it is, how it stains, does it have a flagella or not, what's the arrangement, things like that. How do we classify this bacteria, and where does it fall on the classification scientific scheme of prokaryotes? Well, there's lots of different ways to do this, and it all depends on how you want to diagnose your species. Well, you can do it underneath a microscope. Thought I had those put out. All right, so you can look at it underneath a microscope. That's the easiest way to do it. See what they look like underneath a microscope, see if they have flagella, things like that. See how it's, things like that. You can look at it on a plate. Certain microbiologists come across the same species over and over and over again, and they can diagnose them based on just looking at them on a petri dish based on how they look. You can do that if you're capable of doing it. You can analyze how the bacteria itself does things. Is it capable of eating these particular types of sugars? Can it metabolize this protein? Does it produce this waste product? Blah, blah, blah. All the different characteristics of bacteria that are unique to different species. You can test to see which one it is. You can look and see what the bacteria does underneath testing of the immune system, or coming in contact with blood and samples from human and things like that. Or you can just run its genetics and see what it says that way. So there's lots of different ways to diagnose bacteria and figure out where they are and who they're all related to. Lots of different ways to do this, lots of different manuals to do this, but according to the most commonly used way of breaking out bacteria, there are prokaryotes. There are really 2 main groups. We've talked about these a lot. Archaea and bacteria, with 5 major subgroups and 25 different phyla of bacteria. You can check them out here. There's a little more information about all of those. Don't memorize these charts. So these are just some of the main groups. You've got domain archaea. We've talked about these a lot. These very strange little organisms that live in really harsh environments. They do some extreme things. They eat methane, eat weird things. They live in really harsh environments with very high temperatures, very high acidity, and things like that. Really weird little organisms. And then you have the bacteria that we're familiar with, domain bacteria. Proteobacteria, organisms with gram-negative cell walls. Firmicutes, which are mostly going to be gram-positives with a low G and C count in their DNA. These guys tend to be a little more. G and C count has to deal with how susceptible they are to UV damage. More A's and T's, the more susceptible you are to UV damage, but that's kind of either here nor there. And then Acidomycobacter, which are gram-positives with high G and C content. So they're a little more resistant to UV light. So that's kind of the breakdown of those. You can see here on the universal phylogenetic tree, bacteria are totally separate than Euchara and Archaea. So Archaea and the Euchara over here, bacteria is on the other side. So very odd little organisms, totally separate from us. So how do you diagnose these guys in a lab? You're going to diagnose them underneath your microscopes based on how they look, how their structures and shapes are put together. You're going to base apart how they perform on media, what they do under certain conditions and things like that. And that's how you're going to diagnose these guys underneath a medical condition. Also, it's kind of moved towards DNA, lots of other types of identification for medicine nowadays. Some very important bacteria in the medicine world and that you'll probably encounter if you end up as a nurse or a doctor or something like that. Once again, don't recommend memorizing these charts. Just take a glance at it, but don't memorize them. Bacteria, once you've identified what the kind of overall genus is, you can start to break them down into species, strains, and varieties and types. And you'll hear this a lot when it comes to identifying viruses and things like that, types and strains and stuff. But for bacteria, anyway, living organisms, this is how we do this. So what is a species of bacteria? Well, a species of bacteria is going to be bacteria that are all going to have the very same characteristics or very similar characteristics to one another that differs from everyone else. So every white-tailed deer on the planet pretty much does the same thing, and there's nothing else on the planet that's not a white-tailed deer that does that. And that's why we call it its own species. And bacteria work the same way. They all pretty much do the exact same thing. They all look the same. They all behave the same. So we classify it as a species. It's its own unique thing. Nothing else does that unless you are that kind of thing. Now you can have strains, and a strain is kind of like a kid. They're just slightly different. They're the same, but just slightly different. So a strain or a variety would be like Salmonella, but it has everything about Salmonella, but it can do something different that maybe its father or its mother, its parents, couldn't do. So it's got something slightly different from it. And then you've got a type. And types are a species that genetically are different in the sense of how they express themselves. They're either able to cause disease better or worse. They're more viral, less viral, or they cause disease in a new way or a different way than the original Salmonella might have done. So a strain is something that can eat something different, or it's put together a little different. But a type is going to be something that's going to cause disease in a different way, or have immunity or something about it, so the way that it causes virility that's different. So that's what to keep in mind with that. Then there's some weird little prokaryotes out there that are classified, that are free-living bacteria that are non-pathogenic, like cyanobacteria. Blue-green algae is another name for these guys. You have green and purple sulfur bacteria, fruiting and gliding bacteria. And these guys are going to pretty much make their own energy via photosynthesis. Just some weird little guys out there that you might come in contact with. And they might not even need sunlight. They might just get inorganic compounds to do this. So strange little guys. So cyanobacteria, one of the most common little tiny microscopic organisms on the planet. You can find these in your front yard all over the place in the form of something called nostoc. These guys are hypothesized to what gave rise to most of the oxygen on the planet. Very simplistic form of photosynthesis. These guys arose a very long time ago. They are capable of carrying out oxygenic photosynthesis, the same thing that plants do, in a very similar but very simplistic way. Is about 40% of the oxygen that we breathe on a daily basis. I think it's like 40 to 60 is the estimate, is produced by these type of organisms. Now, if you stain them, they would be gram negative, but they don't behave in the same way that typical gram negatives do, in the sense that they live out in the water, they're photosynthetic, or they live in the yard and things like that. They like moist environments where they can make photosynthetic products. So this is a stromatolite. It looks like one of those fungus that grows on the side of a tree, a branch fungus, but this is actually a rock, fossilized rock, stromatolite, and this is the cross section of it. So it looks like a big rock, and they just essentially cut it in half, slice the cake out of it. And what these are fossilized cyanobacteria that are found all over the ocean. So these guys, as I mentioned, were kind of the first things to evolve on the planet that made oxygen for us, that allowed the organisms in the water to move onto the land as the environment got more oxygen in it. And they would gather together on the seashores around the Earth and form those little biofilm balls that would build up into on top of one another. And that's what this is. They're called stromatolites. They're found all over the shores of the world. Big balls of fossilized cyanobacteria that formed in the very early Earth, that were all kind of a giant blob of biofilm to produce oxygen. Very odd little organisms, but still pretty neat. You have your blue and purple photosynthetic, or blue and purple, green and purple sulfur bacteria. Gosh, I cannot read that today. Now these guys are going to be strange. They're not going to produce oxygen via photosynthesis, so this is anoxygenic photosynthesis. They'll produce that methane and those weird, funky sulfur smells and things in still ponds and things like that, stagnant water. Gliding and fruiting bacteria, very odd little organisms. They just move around from place to place, scoot around, slide around. Most of them are going to be gram negatives. Nothing really of concern here. They're very uncommon in the sense of causing disease for people. Then you have some other weird little forms of bacteria that are forced to live inside of other cells. Now, we've talked about microbial relationships already with parasites and things like that. Now, some bacteria are parasites. That seems very odd for something as very small as a bacterial cell, but some of them are considered intracellular parasites, which means they live inside of another cell. You can either be an obligate or a facultative. Obligate means you have to, facultative means you will if you can, but you don't have to. So an obligate intracellular parasite is a bacteria or an organism that absolutely must live and must be inside of another host cell, either to successfully cause disease or to reproduce or something like that. At some point in its lifespan, it has to be inside of another cell. So there are quite a few of those in the medical world, but one of the main ones that people are concerned with is Rocky Mountain spotted fever, or Rickettsia rickettsii. It's a very, very, very small cell. They live inside of the human host cell. And when they do this, the immune system has no earthly idea they're there because it can't see them. So they're able to cause disease without notifying the immune system that they're there. Chlamydia is another bacteria that's able to do this, another intracellular parasite. Oddly enough, bugs can't get chlamydia. It's kind of strange. Crabs and things like that, but birds and humans and other organisms can. They're very deadly to parrots and birds and things like that. Chlamydia psittaci is their form of this, but Chlamydia trachomatis is the one that causes problems in humans with eye infections and things like that. Most common STD on the planet. You can get it from rubbing your fingers in your eyes and things like that. If you've touched a chlamydia bacteria, babies get it a lot through the process of birth, coming in contact through the birth canal. Different form of chlamydia causes quite a few lung infections that can be fatal as well. So then you got archaea, our last little weird group of prokaryotes, this strange little domain off by themselves. Closer related to us than to true bacteria. And once again, they live in those harsh environments that are very either warm or very high in acids and bases and things like that. So their proteins have to be able to survive in that high heat, high acidic environment. So the structures that make those proteins, those RNA sequences and things like that, are slightly different. And the way that their cell walls and cell membranes are put together are different as well to compensate for those harsher environments. So that's the differences between prokaryotes and traditional prokaryotes, bacteria and things like that. And then our archaea, the other little weird group. So once again, you're going to find these guys in those crazy, harsh environments. These are archaea that cause these fields to turn red overgrowth of archaea. These are water ponds. I think these are fish farming ponds and things like that. You can do this in lots of different places. But our overgrowths of archaic bacteria or archaic organisms can cause toxins to be produced that are fatal to humans, fatal to fish, and things like that. And that's what's going on here. So lots of weird things, lots of weird little archaic organisms produce lots of very strange products. Sulfur, they eat sulfur. Some of them love salt. Some of them produce methane. Weird little guys found in very strange environments. But just be aware that they do exist. They're cool though. That's pretty much all I've really got for this lecture over the anatomy of a prokaryote, how they're put together, and kind of the classification of prokaryotes, and kind of some of the prokaryotes that are out there. So if you have any questions over this lecture, if you just want me to elaborate on anything, or you want to just know something, feel free to send me an email and let me know. If not, have a great rest of your day.